Welcome everyone to NF2 Biosolutions' first webinar, The Promise of Gene Therapy for NF2. We are so happy you could join us. My name is Helene Bader. I am a volunteer with NF2 Biosolutions, and I just want to give a few quick housekeeping notes before we get started. The webinar is being recorded and will be available on our website for future viewing, www.nf2biosolutions.org. All participants will be muted throughout the webinar. You'll be able to ask any questions through the chat function. We will have the chat box locked until after the presentation and then open it up for questions. So please wait until the end of the presentation to ask any questions so it will be easier to get to your question and it won't get lost. And if for any reason we are unable to get to your question, we will put together an answer document and send that out to all webinar attendees in the next few days. And finally, we are planning to have regular webinars throughout the year, and we would love your input on what topics you would like to see covered and what you would like to hear about. So please feel free to enter your suggestions in the chat box at the end of our session along with your questions, and we will compile a list of topics. And now I'd like to introduce our presenter today, Dr. Edgar Rodriguez-Lebron. Dr. Rodriguez-Lebron, Chief Scientific Officer for NF2 Biosolutions, has 20 years of experience in the gene therapy space with a focus on diseases of the nervous system. He has engineered and tested gene-based therapies to prevent or reverse neurodegenerative processes caused by the accumulation of toxic mutant proteins. He is currently the CSO of Andante Biologics and a co-founder of multiple early-stage biotech companies developing molecular therapies for CNS diseases. He holds a faculty appointment at the University of Florida, where he trained as a graduate student. So Dr. Rodriguez-Lebron, take it away, please.
Good evening, everyone. I would like to first give a thanks to all of the folks that were involved in organizing this webinar. It is truly my pleasure to be able to speak to all of you tonight. I must say that this past year, the amount of experience and friendships that I have built through NF2 Bio have truly been inspiring. And I have really, truly enjoyed interacting with all of the NF2 Bio people. You can see we have put up a poll, and throughout the presentation, you will have a number of questions that will pull up, and we'll leave those polls open maybe for about 30 seconds to at least give 50% of you a chance to vote. You'll see some questions that are just sort of general basis, such as the first one: how would you describe your knowledge of gene therapy? And so at this point, you notice we have about 75% of you already voted. So I will go ahead and end the polling so we can initiate the webinar. And these are some of the results in case you guys would like to see. So it looks like a good portion of you, about 70% of you, have already a pretty good understanding of gene therapy. So that should make tonight's job for me quite simple. Okay, so we will go ahead and stop the polling for now and move on to the presentation. So, excuse me for a second. Looks like I'm hosting the presentation, and there we go. So the basics of genetic disease. This is truly where all of gene therapy initiates, right? Is the whole notion that there are a genetics, or that there is a genetic basis for multiple diseases, including NF2, and that we have different methods through which those diseases are inherited. And so what I'm demonstrating here is just two very basic modes of genetic inheritance. One is a dominant form, and the other one is a recessive form. And as you can appreciate, in a dominant case, which is the case for NF2, really all you need is one allele in either an affected father or an affected mother to be passed along to the children. Now, in the case of NF2, it's a little bit different, and we'll get to that in a second. But that is a dominant pattern of inheritance. Only one copy of a mutant gene gets passed along to your progeny. In the case of an autosomal recessive pattern of inheritance, two copies of a mutated allele or of a disease-causing allele needs to be passed along to your progeny in order for disease to manifest itself. And really, since we understood this, very early on, scientists began realizing that perhaps if we could identify really the deficiencies that underlie a number of inherited diseases, if we could identify those genetic elements that were responsible for those disorders, then potentially one could engineer a type of therapy, and they called that a gene therapy. So ever since 1972, when Thomas Friedman and Ron Roblin published in the journal Science an opinion piece, scientists have been thinking about the development of gene therapy strategies. This is all the way back to 1972. And what I'm presenting here is excerpts of that paper. And it's just really fascinating to read what these two scientists wrote back in 1972. In our view, gene therapy may ameliorate some human genetic diseases in the future. They also said, but for the foreseeable future, however, we oppose any further attempts at conducting such therapies. And this was really interesting because they went on to delineate and to argue as to why they opposed any development of gene therapies. They pretty much went straight at the lack of knowledge that we had regarding genetic regulation, genetic recombinations, how we could precisely match genetic defects to actual disease states. And importantly, they also thought about the safety of such a novel approach by suggesting that there was really no information on either short or long-term consequences of using these therapies. And so it's really after the genomic revolution that we have experienced in the last 20 years or so, that gene therapy and that the promise of gene therapy actually became a reality. And so really, it just reminds me of a quote, one of my favorite, I like to call him a scientist, although I guess he's an inventor. When he said, Leonardo da Vinci said, people of accomplishment rarely sat back and let things happen to them. They went out and happened to things. And that's exactly what happened with gene therapy. In the 80s and throughout the mid-90s, there were a lot of failures and a lot of lessons learned. And some of those lessons were very tough. Here I'm showing the picture of Jesse Gelsinger, who actually passed away after receiving a gene therapy treatment at the University of Pennsylvania from an uncontrollable cytokine storm. And out of all those failures, we really, as a field, grew. We became more disciplined and learned sufficiently enough to be able to make gene therapy a reality. And so as you can see now, the inflection point occurred a few years back where now number of successes are really truly now breeding confidence into this therapeutic modality. And so that confidence has really spawned a revolution in medicine, what we like to call gene medicine. And what you can see here in all these spots are all gene therapy based drugs that are on their way to approval by the FDA. And these are all the different areas of disease: hematology, dermatology, metabolic diseases, ophthalmology, and oncology, which are all now being targeted using this therapeutic modality. And I would like to just highlight probably one of my favorite success stories. Of course, me having a background in neuroscience, this one hits very close. And in fact, I was very close to the early stage development of this drug when it was first conceived by Dr. Brian Casper. And this is a story of spinal muscular dystrophy. As you guys know, it's a pediatric disease. It is caused by the loss of expression or the loss of function of a protein called the survival motor neuron protein. And just as its name implies, this protein is essential for the survival and function of motor neurons. So when you lack this protein, your motor neuron units begin to degenerate and you begin to lose all spinal cord activity and function. One of the main problems in spinal muscular atrophy was the notion that even though we knew what the missing gene was, and we knew that that's the gene that we had to replace, the barrier was, how do we deliver that gene into the cells that need it, meaning those motor neurons that reside nicely tucked away and protected in the spinal cord. And really, it was after years and years of work and somewhat of serendipity that Brian was able to find that a particular virus, which also happens to be non-pathogenic to humans, was capable of effectively crossing that blood-brain barrier and reaching those cells in the spinal cord to deliver a genetic cargo that included the SMN gene. And it's really quite a story, as I can just show you here very quickly. When you look at the natural course history of this disease, only 8% of children at the age of 20 months have or show no disease. And I'm not even going to describe the phenotype of this disease, but it's pretty devastating. Now, what I'm showing you here are results from the first studies using this AAV gene therapy drug, showing how at 20 months of age, 9 out of 9 of the kids that had reached that age all were event-free. And at 13 months of age, where 25% of the kids tend to be event-free, 100% of the kids treated were event-free. And so these results were just so beyond what anybody expected, that it really immediately led the FDA to approve this drug for the treatment of SMA. And it was really because of the amazing efforts of not just Dr. Brian Casper, but also Dr. Jerry Mandel at the Nationwide Children's Hospital. And, you know, one, I guess, takeaway message here that is important for us to understand is that it's not just about designing the drug, and it's not just about designing the gene therapy. We have to have a multi-pronged approach that includes our ability to truly understand the natural history of disease and to be able to identify and utilize reliable biomarkers of disease course. Because especially in the cases of rare diseases, to be able to properly design the clinical studies, having this information in hand truly helps the FDA be able to guide the process all the way to approval. And that is really a long process. It's a process that initiates at the time when you first conceive an idea, identify a gene or genetic defect that you would like to go after or target using gene therapy. It then follows your design. How are you going to express this gene? When do you want it on when do you want it off if you need it off? How are you going to reach those target tissues that you need to reach? Once you have that design, then you need to move on into testing this in animals and make sure that your approach not only works, but that it is safe. Then after that, you have to look at it in actual models of the disease that you're targeting to show that it's not just safe, but that it actually has efficacy. And so forth and so on. You can see that this is a long path that eventually leads to the second half of the development phase, which is clinical studies. And so all of this that I'm highlighting here is what we refer to as non-clinical development, which is typically in a gene therapy program, a path that can take many, many years. And after that, then you move on to clinical trials, which can also take a number of years before completion. And so all of gene therapies, no matter what gene therapy you're talking about, all of them have their basis on what we call the central dogma of molecular genetics. And my apologies, I know it's Friday night and most of you probably want to just have a drink, and I hope you do have a drink in your hand right now. But we're going to do a little bit of school time right now. So what is the central dogma of molecular genetics? It is the notion or the idea that cellular function is completely dependent on what we like to think of as a simple process, and that is DNA, which is transcribed into messenger molecules called RNAs, which are translated into proteins. And that process occurs in all of our cells at all times. Any disruption to that process, such as by the existence of mutations in genes, can then result in, for example, a loss of the ability to create RNA, which would result in the loss of our ability to produce a protein. And we refer to that as a loss of function. So those are mutations that are causing a loss of function at the genetic level. You could also have those mutations not disrupt the transcriptional process, but instead be incorporated into RNA molecules, which then translate those mutations into proteins. Now, those proteins carrying those mutations can, as a consequence, not be able to perform their natural function. Therefore, you will again have a loss of function. But there's also the possibility that those mutations can actually turn that protein into something that it was not engineered to do. And it could be that it could lead to what we like to refer to as a gain of toxic function. All of gene therapies and cell-based therapies are designed by first investigating and understanding which of these two is actually at play in the disease that you are targeting. If the disease that you are targeting is due to a loss of function, then this is a very simple approach. All you have to do is design a gene therapy that would replace that function. And you can replace that function by introducing a healthy copy of that gene. You can also block the expression of a toxic gene, and you would want to do that in the case of a gain of toxic function. Now, things are not always black and white, and sometimes it is the case that mutations can both lead to a loss of the natural function and a gain of a toxic function. At that point, you can now imagine that your strategy would have to address both the loss of the natural function, so you would have to replace that function, and you would have to also address the gain of toxicity by blocking expression of that endogenous toxic protein. Lastly, gene and cell-based therapies don't always have to target the gene that carries the mutation. Oftentimes, we can simply use gene or cell-based therapies to modify the physiology of the organism by affecting specific cellular pathways. And these cellular pathways can either be enhanced by the gene therapy or they can be suppressed by the gene therapy. When we think about gene therapy in the context of NF2, you can now imagine we want to think about the NF2 gene, and we need to understand what those mutations are doing to the NF2 gene, and we need to understand then how we design therapies to counteract those disease-causing mutations. And one of the first things that you will notice when you start thinking about the NF2 gene is that there are many mutations that occur in many different regions of the protein. That's what's being highlighted here. Because there are so many mutations that are affecting so many patients, it is very difficult to conceive of a strategy that would simply be designed to target one of those mutations. Instead, one needs to then immediately think about a strategy that would circumvent the diversity of mutations that could be present in the patient population. It is also important to understand the function of NF2 in order to address whether we are facing a loss of function phenotype or a gain of function phenotype. And one of the immediate things that you will notice and that you will realize is that merlin, or the NF2 protein, is involved in so many aspects of cellular homeostasis, but it is primarily functioning in pathways that are extremely important to cellular division. And so we know that one of the main functions of this protein is that of a tumor suppressor. And just like a number of other tumor suppressors, if you have too much of that protein, that may lead to disruptions in normal homeostatic pathways. And if you don't have sufficient amounts of the protein, you can also lead to disruptions in cellular pathways. And so when we think about the genetics of this disease, which again need to be understood in order to design therapies to target that are based on genetic elements, we know that as a tumor suppressor, NF2 arises from what is called a second hit. So when you inherit an NF2 or a mutation in your NF2, you're typically in a heterozygous state, meaning only one of your alleles is carrying the mutation. When you are in that state, and any cell in your body that has this genetic makeup, and specifically any Schwann cell in your body that has this genetic makeup, performs its normal function as far as we understood. Schwann cells are cells that ensheath the axon of neurons, and so they act almost like the coverings on an electric cable. Their primary function is to modulate that electric signal that travels down the axon and that allows neurons to communicate with one another. This is a little bit of a zoom look at what happens to a Schwann cell after it interacts with an axon. It wraps itself and it forms what is called a myelin sheath. In fact, my early beginnings of science, when I was an undergraduate a long time ago, was to actually look at cultures of Schwann cells with motor neurons, with sensory neurons, CRG neurons, to understand how the Schwann cell wrapped itself around the axon. That is actually how I began science, or my scientific career. But I digress. So what happens in the case of NF2 is that eventually the second allele also acquires a mutation that leads to its inactivation. So you lose the activity of both copies of merlin or NF2, and that eventually leads to the formation of a tumor. So in summary, we know that in the NF2 problem, there is the inheritance of one allele that is carrying mutations, but in this genetic state, cells appear to tolerate these mutations and appear to, for the most part, function properly. Once you inactivate the second allele, that then leads to a loss of function phenotype. And because it's a loss of function phenotype, one could circumvent that by replacing the NF2 copy with a healthy copy. One can also think about potentially not adding an extra healthy copy of NF2, but instead editing the faulty sequence that exists in the body. Lastly, one can also think about modifying specific cellular pathways, as I mentioned before, that could potentially make the tumor vulnerable. One of the main challenges of gene therapy is how we accomplish delivery of the genetic material. In this slide, I'm just simply introducing you to the basics of how one could deliver genetic material into the human body. One can utilize chemical formulations, primarily based on what primarily are lipids, also referred to as nanoparticles, which encapsulate pieces of DNA or whatever genetic material you would like to include. And these entities, which for the most part are synthetic chemical entities, can be infused directly into tissue space or intravenously to let them spread throughout the body and deliver the genetic material. These are called non-viral approaches. There are also viral delivery approaches that take advantage of the natural ability of viruses to infect cells in our body to deliver a genetic material. There's also what we call cell-based therapies, and those are ex vivo therapies, in contrast to these systems, which are called in vivo systems. In the ex vivo case, the cells are isolated from the patient, modified in the laboratory, and then infused back into the patient. And we're going to look at some examples of that. As we think about viral delivery, I would like for us to talk a little bit about a particular vector which has shown incredible safety and efficacy in the clinic. And it is in fact a vector that was used to develop the gene therapy for spinal muscular atrophy, and that is the adeno-associated viral vector. AAV, as we all know it, is a member of a DNA family of viruses, and they're called dependoviruses because by themselves they're incapable of replicating. They need or depend on another virus in order to replicate in the human cells. These are small non-enveloped DNA viruses, so they encapsulate the DNA using an actual protein capsid. The AAVs were initially isolated from an adenovirus infection, and that's because AAVs tend to replicate in the presence of adenovirus. So it was very easy to detect it when analyzing tissues that were infected with adenovirus. What's really very, very important here is that for about 50 years now of observations, we have really not been able to detect any known pathogenic mechanisms that lead to human disease that are due to a natural infection with AAV. That is to say, this virus appears to be non-pathogenic to humans. It also has limited immunogenicity, meaning your body reacts to it, but you would not even notice that your body had actually mounted an immune response to a natural AAV infection. What I'm showing you here is a recent publication from the laboratory of Stephen Gray, who is one of the leaders in AAV gene therapy development. And what he is looking at is trying to understand whether that AAV9 capsid serotype that is currently being used in spinal muscular atrophy can actually work for peripheral neuropathies. And why is this relevant to us is because it's given us an insight into how well some of these vectors can infect Schwann cells, which are going to be our target cells. So when you think about AAV vectors, I would like to tonight talk to you about an analogy which focuses on the space shuttle. Me being from Florida, I'm a huge fan of NASA and all of the period when we could get to go to Cape Canaveral and watch astronauts lift off in this shuttle. And that's really how I think of AAV. It is like a shuttle that is allowing us to deliver genetic cargo into a variety of cells. In this case, what we're focusing on, which are Schwann cells. As I mentioned before, the DNA, which would be the cargo, is delivered in this shuttle. And this shuttle, just like the NASA shuttle, can be designed in many, many different ways. One can modify these capsid amino acid sequence to make these viruses be more prone or less prone to infect or to infecting certain tissues. So for example, let's say you're designing a therapy and you would like your cargo to be exclusively delivered to the heart. You could facilitate your ability to deliver to cardiomyocytes or the myocardial tissue by engineering specific sequences within the capsid shell until you find variants that would effectively deliver primarily into the heart, even when infused into the bloodstream. And why is this important? And I think this is an important message for all of you. It is because in order for a gene therapy to work, we must be able to deliver the cargo effectively and safely to the target tissue. If you are not able to do that, then gene therapies do not work. And this is important. And that is why now there are efforts to use artificial intelligence and machine learning algorithms to look at every single possible combination of amino acid sequences throughout the capsid of this virus. And by altering these every single one at a time, you can generate trillions of variants of these capsids. And you can begin to understand how specific changes to the capsid of this shuttle can either facilitate the transfer of genetic material to a particular tissue or block the ability to transfer genetic material to a tissue which you do not desire to receive the therapy. So as we move on, AAV vectors, as I stated before, are complex biological drugs. And so I described the shuttle, which is the capsid, and that shuttle performs a function, which is to deliver a genetic cargo, and that's the genetic material that we have to put inside of the virus. In the case of AAV, it's very, very, very good for us because only 5% of the endogenous viral genome, that is to say, of the genomic sequence that exists in nature for this virus, only 5% of that remains in the therapeutic vector. So the rest of the viral genome gets gutted out and replaced with the genetic elements that are required to achieve the therapeutic goal. And typically, and this is shown here in a simplistic way, what you would need is a therapeutic gene. In our case, for example, it could be a healthy copy of NF2 and you need a promoter. That is to say, a genetic element that does just that, promotes the expression of that therapeutic gene. And in the same way that one can modify the capsid to try to target specific tissues and specific cells, one could also use specific promoters that would drive expression of the therapeutic gene only in particular cells, or at least preferentially in particular cells or particular tissues. So you can see how there is a two-level or a two-layer approach to engineering the vector. One is the capsid and how we can make it go in the direction that we would like it to go, and also the genetic elements that it contains so that we can control where it expresses that therapeutic gene and maybe even the levels at which that therapeutic gene is expressed. In the case of AAV, one of the constraints that we have is that it has limitations when it comes to the size of how much DNA or the genetic cargo that you can put inside of that capsid. Thankfully, in the case of NF2, this is not a problem. The gene for NF2 fits just fine within the AAV capsid, and it really allows for scientists to play with a number of promoters to determine which ones would be more efficacious in a therapeutic application. So I spent a little bit of time talking about AAV-based therapies, and I just wanted to briefly mention these advantages. It provides really an effective way to enter target cells, in our case, Schwann cells. It is easily engineered to achieve trillions of options in terms of its capsid. It also, which is very important to a number of gene therapies, is capable of facilitating sustained expression of the therapeutic gene. Now, some of the disadvantages of using these approaches are, for example, the manufacturing of this drug. Because it is a biologic, it is a process that requires some time, requires some troubleshooting, and it is a process that is not as simple as synthesizing a molecule. We also have to always be aware of the fact that this is a live virus. And thank God for this, but we do have an immune system that is capable of recognizing these type of pathogens. And even though AAV is not a pathogenic virus, our immune system still sees it as a foreign agent and it tries to neutralize it. So we have to engineer our way out of that trouble. What is important also for us is the clinical track record of AAV. Pretty much no adverse events to date that are major in nature. There are some events that have been reported, but nothing major has been reported to date. So now that I've talked to you about gene therapy approaches, a little bit about NF2, and I've also spoken to you about how we can apply AAV therapies to NF2, I want to get back to this question about delivery. And in the case of NF2, we really have two choices. We can either try to directly deliver the genetic material into the tumor tissue, and that, even though from the surface looks like the easiest, more direct route, it does pose some challenges. As you probably are aware of, even if you are able to treat one tumor in NF2, there may still be a second tumor that arises somewhere else. In that case, you would have to think about how can you design gene therapy strategies that would allow for repeated dosing of that vector. It is not an impossibility, It is just something that we have to think about on how we tackle that challenge. The other way of delivering the therapy would be systemically, that is to say, to infuse it into your bloodstream and allow it to run its course throughout the body. It is challenging because the idea behind this approach is that you would have to deliver your genetic target to as many cells as you can, in this particular case, to as many Schwann cells in your body as you can. And that is no easy feat. Now, is it possible? Yeah, there is some data that tells us that we can achieve effective systemic delivery of a gene therapy. And I'm not going to go into the details, but you guys can look up Sarepta Therapeutics and look at their trials on Duchenne muscular dystrophy, where the problem is in muscles, specifically skeletal muscles. And this company has an AAV-based approach to deliver a missing gene throughout the skeletal musculature of the bodies, primarily in children. So the other thing that we have to think about in NF2 is the idea of, do we replace the gene or do we target the tumor growth? As I mentioned before, one could either replace the loss of function by adding a healthy copy of the NF2 gene, or one could think about editing the existing sequence. You can also think about modifying specific cellular pathways using a gene therapy approach to get rid of that tumor. And I would like to just briefly highlight the work of Dr. Gary Brenner. He is an associate professor at Harvard Medical School and really quite a scientist. I am so impressed by the work that he has done, and really, truly his commitment to NF2. And I'm just so proud that NF2 Bio supports his research and that he is working with us in this endeavor. And one of the things that I just wanted to highlight briefly is the fact that Dr. Gary Brenner has a xenograft model of this disease where he can take cell lines that are derived from human material, and by injecting these tumor-inducing cell lines into the sciatic nerve of mice, he can watch over time how these tumors form in the sciatic nerve. And so this model allows you to really track the growth of a tumor over time. It is based on human-derived cells, and it also leads to some behavioral phenotypes. And I will show you some data on that right now. Really, what's amazing is some work that he has recently published, where he showed that he can actually design an AAV vector with a particular promoter expressing a particular gene. We don't need to get into those details right now, but suffice to say, this gene is designed to induce cell death only in the tumor cells. And so what you see here is when Dr. Brenner induces the tumor formation in those mice and then injects the AAV vector 3 days following the implantation of the tumor-inducing cells, you can see that if he uses a vector that does not express the therapeutic gene, you can see how lack of expression of a therapeutic gene simply leads to the growth of a tumor, which is shown here by this graph going up. In contrast, if you have the therapeutic gene, you can see how the progression of that tumor is halted. And he has seen this in a number of settings, using a number of controls, and using also a number or a variety of different tumor lines, whether less aggressive or more aggressive forms of that model. As I mentioned before, he can also measure whether there's an impact in the actual function of the nerve. And as you can see here, in the case where there is a therapeutic gene being expressed, which is a gray line, the mice are able to recover their ability to sense a change in temperature, a hot temperature, versus animals carrying the mutation or the tumor, but expressing a vector with no therapeutic gene. Also here, you can see an actual mechanical phenotype where the animals lose their ability to be sensitive in this assay and then recover if they're expressing that therapeutic gene. This program, we like to call the AAV ASK program, is now in non-clinical development stage, and it is something that we are currently focused in facilitating and helping Dr. Brenner move expeditiously into the clinic. One question that we have, however, is this is working in a model where a tumor is being treated in a sciatic nerve setting. Could this work when we think about vestibular schwannomas? Another approach, as I said, is one could either target the tumor, but one can also try to replace with a healthy copy of the NF2 gene. And that is what's called a gene replacement strategy. And work that is currently being performed at Nationwide Children's Hospital. Remember, that's the site where the spinal muscular atrophy program was developed, and in fact was developed by Dr. Casper. Work right now being done by Dr. Meyer, who trained with Dr. Casper, is focused on trying to optimize delivery strategies and trying to evaluate the efficacy and the targeting of the tissues using an AAV vector approach that expresses a healthy copy of NF2 merlin. And as shown here on the right, the idea would be to replace the lack of function with a healthy copy of the gene. And Dr. Meyer and her team are working very, very hard at looking at a number of different AAV capsids and a number of therapeutic expression constructs. Yet another approach that we discussed before is the notion that you could also replace with a healthy copy and at the same time eliminate expression of the mutant when it exists. And that is an approach that we refer to as an ablate and replace approach. And that is being conducted by Dr. Chris Mueller and his postdoc, Dr. Meijboom, who are testing this approach, again, first by designing AAV vectors that in this case would deliver two molecules, a functional copy of the NF2 gene and also a molecule that can target the destruction of the endogenous NF2 protein. To end, I would like to just briefly mention a little bit about cell-based therapies. So far, we have focused on viral-based approaches, which are an in vivo based approach. That is to say, the patient will receive a viral vector that was manufactured under quality control conditions to be infused either directly into the tumor or intravenously. One could also follow a different approach called the ex vivo approach, in which we can isolate cells from the patient, modify them ex vivo, that is to say in the laboratory, and then infuse them back into the patient. And you have probably heard of gene therapy approaches that have now been approved, which are referred to as CAR T-cell approaches. And what are CAR T-cells? And I apologize that this picture did not come through as well as I would want it to come through. But briefly, the way that a CAR T-cell works is you would isolate cells that are part of the immune system. And what you're trying to do is basically hijack the immune system and reprogram it so that it can now generate immune cells that can hone and target cancer-bearing or cancer-causing tumor cells. Now, this approach has shown great promise and has been very effective in some forms of blood cancers. And those are different than NF2 in that NF2 is due to a solid tumor mass, which is not the case in situations like leukemia. And so one barrier that exists between the use of cell-based therapies and an effective NF2 therapy is the fact that these cells tend to not perform as well when faced with a solid tumor microenvironment. And so a lot of effort is now directed towards redesigning and re-engineering these type of approaches so that they can work effectively and efficiently against solid tumors. If you think about this, approaches that are based on immune cells, if effective against solid tumors, would really be a game changer. Primarily because the main challenge of a viral vector approach is our ability to deliver throughout the body or effectively in the tumor. Well, in the case of cell-based therapies, we don't have to worry about delivery because those cells are already naturally evolved to pretty much exist everywhere in our body. So definitely trying to engineer these approaches to be more effective against solid tumors is something that NF2 Bio is focused on. Also understanding how we can engineer these cells to be able to limit some of the toxicities that are known to be associated with these treatments. As our first effort in that enterprise, we have now partnered with the Children's Brain Tumor Tissue Consortium, as well as with WuXi NextCode, to not only collect NF2 tumor material, but sequence the genomes of these tumors, perform sequencing of gene expressions, and by using artificial intelligence algorithms, try to hone in on signals that are specific to NF2 tumors. By identifying those signals, one could then begin to develop T cells that are engineered to latch on to NF2 tumor-specific antigens. Another aspect of this collaboration, which is important for our mission of developing a gene therapy for NF2, is the fact that it would help us better understand what signatures can we follow and what signatures can we measure once gene therapy approaches enter clinical studies, so that we can understand whether those approaches are being effective, and so that we can understand which way can we improve on those therapies if the efficacy falls short of our expectations. So with that in mind, I would like to simply summarize by stating that viral-based therapies provide an opportunity in NF2. Primarily, it gives us multiple avenues to target this disease by either replacing the faulty NF2 gene or by targeting the structure of the tumor tissue. We have to focus on the delivery aspects of these diseases, of these vectors. So delivery, delivery, delivery. We have to also think about how we are going to manage any potential immune reactions to the vector in the case there is a need for redosing. But really what we are so excited and hopeful about is the fact that the path has been laid out. There are already pioneers who have shown us how we can get to our goal. And it is in building and standing on what we like to call standing on the shoulders of giants, that we can take projects like Dr. Brenner's AAV Ask project and quickly move it down the path to clinical phase studies. Other gene therapies that are going to be targeted to the NF2 problem are also continuing to show promise. Cell-based strategies, as I said, have shown now some evidence of improved efficacy against solid tumors, and we are, NF2 Bio is prepared and building all the foundation that it would be required to quickly adapt some of these strategies into the NF2 space. Other approaches that NF2 Bio is really facilitating and paying attention to involve the use of bacteria. Are bacteria coming to our rescue? Can engineered bacteria not only kill NF2 tumors when infused directly into the tumor space, but could they also block formation of new ones later on? Can we use the bacterial immune system, known as CRISPR-Cas9, also a gene editing approach, to open up a new front on this war against NF2? And so I would like to thank you for your time, and I want to try to leave you with this video, and I will try to see whether the sound works. It was giving me a little bit of trouble before, but I hope you can enjoy it.
One of the most valuable things that NF2 Biosolutions is doing is taking families, clinicians, and scientists out of their silos, bringing them together. The only way we're going to optimally advance towards better treatments and a cure is by working together, by understanding each other, and through a global collaboration focused on one goal.
And we have now been able to establish clear research collaborative efforts between gene therapy labs that would have never looked at the NF2 problem and researchers who have completely focused all of their scientific career in the problem of NF2. And it is in that merge that now NF2 Biosolutions is promoting, facilitating, and in some instances even providing the financial means for some of these new collaborative efforts to move forward.
That really helps keep us motivated and keeps us focused on what we're here to do. We're here to understand the disease and use that knowledge to develop interventions that would benefit patients.
We can give these patients a normal life in terms of quality of life and life expectancy, and take that burden of the fear and the psychological elements of not knowing when the next bad tumor is going to arise out of their lives. We need better treatments, and we need them not tomorrow, but today.
I need a cure.
Okay, thank you so much for your time. I think Nicole Henwood, our president of NF2 Bio, would like to share some words with you guys.
All right, let's see if this works. Can anybody see me?
I can see you.
You can.
You want me to stop sharing? Hold on.
You might need to stop. Yeah, stop sharing your screen. Okay.
Right.
All right. Wow, Edgar, that was amazing. Hi, everybody. This is Nicole. Nicole Henwood. I'm the founder of NF2 Biosolutions, and I'm going to wrap up with a couple words tonight. First off, hats off to Edgar. That was wonderful. You broke it down in such a great way, and thank you for that. I can tell you put a lot of work into that, so thank you so much for this and for everything else that you do for us every single day. And thanks to all of the participants. Looks like we have quite a few people here, so I'm thrilled about that. Thank you for spending your Friday night with us on World NF2 Day. So I just want to wrap up with a little bit of exciting news. Edgar gave a little bit of a teaser for us that bacteria are something that we think might play a really big role in NF2, and we want to announce a new collaboration. We've talked a little bit about Dr. Brenner's work, and we have a new collaboration. He and Dr. John Michelanos, also at Harvard, the chair of microbiology, have teamed up to start a new project for us, and you're going to hear a lot about it over the next couple months. We are kicking off a new campaign to support the bacterial therapy today on NF2 Day, and over the next 2 months, we're going to raise the funds to support that project. But just a quick little summary about that project. We have some really interesting data showing that bacteria can shrink NF2 tumors in the mouse model that Edgar showed us a little bit ago, the xenograft model. And there's a few ways that this happens. The bacteria can directly kill tumors themselves. They can induce that very special immune response that Edgar told us about a little bit as well, the cells that live naturally throughout our body, and can draw in those cells into tumors to start chewing them up, basically. And it can also, they can inhibit blood vessel formation, and tumors, as we know, are dependent on blood vessels. So we have the work that Dr. Brenner has already done that I just told you about, and now in association with Dr. Michelanos, they're going to arm these bacteria with some special weapons that we will be telling you more about over time and add to the power of these bacteria. So we're really excited, and we're going to be sharing that over the next couple months, and we'll have some webinars devoted just to that project, and are excited to bring you all into that project as well. I think we were going to answer some questions now.
Yes, I know that there are some questions. I see, I think 3 questions up at most. And I think what I'm going to do, since all we received were these 3 questions, is I will go ahead and answer these right now. So actually, out of the 3, 1 is probably not, well, actually, people are telling me to share my screen to be able to see the, hold on one second. Sorry, guys. Obviously, there's only so much one can learn. Okay. Does everybody now see, or can somebody at least answer to me? It doesn't matter. Do you see the closed caption again?
Yes, we see them. Thank you.
Yeah.
And do you see the questions? Or am I the only one seeing the questions?
No, we can't see the questions, but why don't we just read the question?
Okay.
And then,
so the question is, one question was, if we just target Schwann cells with gene therapy, how will this help NF2 patients with multiple different types of tumors? So as far as we understand, and this is a very good question, most, if not all, tumors that are associated with NF2 arise from Schwann cells and are due to the transformation of these cells away from their fully differentiated state into a tumor-like or into an undifferentiated state. So what we have to, oh, hold on one second. The reason there's now a number of, my apologies. There's a number of people talking at the same time, and now everything just popped up. Okay, so let me just finish this answer. So as far as we know, delivering into the Schwann cell and expressing into Schwann cells should work for most of this. There are some instances, obviously, in the case of meningiomas, where the delivery strategy may be a little bit different. Regardless of whether we are delivering to Schwann cells or we have to redesign to deliver to other cells, I think the overall strategy of testing whether some of these genetic cargoes will work to arrest the growth of the tumor is really important. So for example, if we are able to develop an AAV based therapy that proves effective in the case of vestibular schwannomas, we could potentially quickly turn that same vector around to then try to target ependymomas and maybe meningiomas. Another question was, does limited immunogenicity in AAV vector mean that you would not become immune to the virus if injected? I'm wondering basically if a patient could get that vector to deliver a type of gene therapy that, say, targets a particular type of tumor once, and then years later could get AAV vector to deliver gene replacement therapy. I'm not sure if this question makes sense. The question makes perfect sense, and it makes me see that you understood the point that I was trying to say. So one of the approaches, or one of the aspects to an AAV gene therapy that one needs to be vigilant of, is how the immune system responds to that therapy. And there are basically 3 parts to that response. First, you have to look at the status or the immune status of the patient before you even deliver the AAV vector. And you're trying to determine and to look to see whether that patient has, for example, antibodies that could recognize that AAV vector already present in their bloodstream. So that's the number one thing you have to look at is, are those patients in a pre-immune status? The second thing that you would have to look at is you have to monitor what the response is when you infuse the vector. And so you are monitoring the patient's immune system for the production of antibodies against that therapeutic vector. And there are protocols using existing immune suppressant drugs that really help in trying to taper down or block the response to the infusion of the AAV therapeutic vector. Now, the third thing is, I think what your question is referring to, which is what would happen if you become immune and then later on you want to receive either a second dose of the vector or a different version of that vector. And again, this goes back to the notion that all that clinicians are currently focused on is trying to make use of knowledge that has been gained from transplant, tissue transplant efforts, right? So understanding how to immune suppress patients so that they don't reject transplants of tissues, taking that biology and applying it to the gene therapy space. And so as of today, there are no AAV therapies that are designed for a redosing event. Most of these therapies are designed for a one-time dose. But there are a number of companies and a number of efforts, both in the industry and in academia, at designing and engineering ways in which we could redose patients with different AAV vectors. There's a question of how long before we can get to clinical trials. That is always dependent on the development stage of the programs. We are very hopeful that some of the programs that we discussed today could advance to clinical trials in a somewhat relative short time frame. And by that, I mean, no more than 5 years from now. As a patient of NF2, what is the most important thing we can do today to help support your research and work right now? The most important thing that I think you can do, and Nicole can probably say some words on this as well, is I think spread the word, make people aware, and try to find as much support as you can within your community. towards this cause, number one. Number two, stay in contact and stay in touch. Sometimes there are needs, like I listed, for example, our efforts towards collecting tumor material so that we can understand better the signatures for some of these tumors.
Yep.
Go
I was
ahead,
just
Nicole.
going to chime in and say that the tumor bank is something that as a community we can rally behind, and the more samples of tumors and varying tumor types that we have, the faster and deeper we can get to an understanding of how this disease works and how we can target it. So keep that in mind. Anybody who has frozen tumor from prior surgeries or knows anybody or who is themselves planning to have surgery, please get in touch with us. We have a great team at CBTTC, and they will make it very easy for you. Just give them a call and they'll take it all from there. So if anybody wants more information about that, please do send us a message.
More questions are coming in. There's another question asking, how do we engage more scientific communities and stakeholders to take interest in NF2? Well, I don't know if, I mean, obviously today is May 22nd. We know it's, NF2 Awareness Day. It is important to really, discuss this, talk about it, and be able to, even within your local communities, reach out to your academic centers. Look for people who may be involved in, whatever gene therapy projects they may be working on, or, whatever other type of tumor or cancer they may be working on, and try to convince them to look at NF2. It is always very good to have as much diversity as possible, because that is how you come up with the best ideas. Who determines the ethics about gene therapy clinical trials? For example, will children be eligible for gene therapy clinical trials? Is that determined by the FDA? Correct. That is determined by the FDA. As particular programs move in front of FDA panels, which are the ones in charge of guiding the development, a number of decisions are made regarding safety first. And so in certain indications such as NF2, there's going to be likely a requirement by the FDA that safety is first demonstrated in non-pediatric patients, and once that safety is established, then those trials can then move into the pediatric population. What is the greatest challenge you foresee to getting a gene therapy? I think the greatest challenge in the case of NF2 is to be able to demonstrate that if we are going to follow a systemic delivery, that we can actually effectively target not just tumor cells, but also the natural Schwann cells, so that we can try to limit the therapy to a single dose. That's going to be very challenging. Now, in the case of strategies such as the one that Dr. Brenner is developing that targets the tumor directly, I think that really the greatest challenge is to achieve effective engagement of that target within tumor cells, and be hopeful that those tumor cells don't figure out ways of circumventing that challenge that the gene therapy will pose to them. But we feel comfortable that some of these approaches are going to work quite effectively. Can stem cells harvested at birth be used unhelpful for this research? That is definitely outside of my comfort zone, but I'd be happy to. I'm going to mark your question, and I'm going to try to find an answer to that. So my email, you can probably access it at the NF2 site, or if you can just send that question again to someone at NF2 Bio, we can try to find an answer to that. Unless somebody knows how stem cells harvested at birth be used or helpful for this research. We can, there's a number of things that can be done with stem cells, right? You can develop cell lines, you can try to differentiate them into Schwann cells, but how you can then turn those around to, use them in the type of gene therapy approaches that we're developing right now, is not necessarily something that is clear at this point. hi, this may seem like a, well, I'm not going to say that word, but it's seems like, okay. So they're asking a question, and the question says, when will we get a viable gene therapy for NF2? Are we 10 away, 15 years away from having gene therapy as an option that can replace or complete surgery or chemotherapy? That's a tough question. And so I am not going to venture to answer that question. We are hopeful that we are less than 10 away. And so with hope, we work full of energy. As you can see, I'm here on a Friday night late, talking to all of you. So we are committed and hopeful that we can try to get this done in less than 10. And that's what we're working for. That's our goal, and that's our mission. But if I knew exactly when this was going to occur, man, I would probably be a very wealthy person. Okay, last, what advice do you give to NF2 families to give them hope? Nicole, I think that sounds like a question for you.
I knew you were going to do that. Well, I think the fact that we're here in less than 2 years, this has gone from a concept to an international organization. We are, happy to announce we just got approved as a charity in the UK also. And an official charity. We had always had an unofficial chapter there with some really amazing team members, but now we're recognized by the UK as well, which allows donations to come in from all of Europe too. So it's the speed and the trajectory that we're on, I think should give hope to everybody. And, so I have days where I feel like there's not much hope myself. And then I remember all of this. And just if we all stick together in this and follow the path of some of these other trailblazer rare diseases like SMA that Edgar told us about, we could be the next SMA. But that community, I have to say, really stuck together. They went door to door to every pediatrician's office, dropped off literature, contacted every family, and really rallied behind the disease. And if we do the same, we could be the next SMA.
And I would like to add to that as I said at the beginning of the webinar, my experience with this group has been just nothing short of amazing. Not just what Nicole does, but really everybody, from Gills to all the other people who are involved in this. Even the folks that were extremely helpful in setting this webinar today. And really, it's when you put this much effort and you put these teams together, you can get a lot done. And even one of the last slides that I referred to with this team that Nicole has able to put together between Wuji Next Code and the children's brain tumor from CHOP, really that team. It's really quite impressive. I mean, I wish all of you could be in these phone calls when we sit down and we discuss how passionate people are about NF2. I mean, I just wish you guys knew the amount of passion that there is out there that exists of scientists and people who are not directly impacted by NF2, who want to participate and work with us. And they're particularly impressed by how committed we are as a group to move a therapy forward, and really very, very impressed by the amount of patient advocacy that exists in this organization. And I just wish you guys were really present there to see how those conversations go. We have people in California waking up at 6 a.m. to have meetings about NF2, right? To discuss how we can move some of these projects forward. So there is a lot of energy, there is commitment. Now it's a matter of matching that energy with resources that can help us move some of these projects, to with safety, but effective speed. Okay, so I think we will call it a night, if that's okay. Again, it was a pleasure being with everybody tonight. Thank you so much for your time. As they stated at the beginning, Helene, I don't know if you want to say a few words. I definitely want to thank you and John and Liz and Jill, sorry, for all your work and trying to put the webinar together. So if you can turn your mic on and you know, take a thank you and a little bow from us.
Thank you.
Yep.
And thank you for a great presentation. And we will follow up with a recording of the video that we will send out to everybody. And we will also make sure all the questions were answered, and we will put that in a document as well. And we will make sure the entire video is captioned, so you'll be able to see that as well. So thank you so much, and have a great evening and a great weekend. And please visit www.nf2biosolutions.org for any additional information, or you can find us on Facebook as well. Okay, take care, everybody. Good night.